hopefully you've already, there we go. Look at this. I've got serial dilution practice questions. I found a good video that goes through inducible and repressible operon so you can see the trip and the arginine operon questions. Question? Okay. More serial dilutions on how to do that. You're going to expect 1 or 2 questions on serial dilutions. Don't spend 6 hours on it. Okay? If you're having trouble with metrics, you're probably going to have a little challenge with this too. Do the videos. Go step by step by step. We don't have time in lecture to do that. If you know that's something you're weak on, make it strong. Okay? All right. How to increase microbial count, generation time, and microbial growth requirements. So as we go through this, and you're like, well, I'm still not sure, start there. Okay? Is that cool? Okay, so off we go. Here's microbial growth. We've already been through binary fission. When we talk about generation time, it's the amount of time it takes for a microbe to double. What's the average generation time for microbes?
17 minutes.
Yeah, 17 minutes. Yeah. Okay, good. Remember, it's exponential. Okay, you've seen this in lab. You've seen this before from the first week of class. I'm not going to spend a ton of time on it. Okay, this gives you a mathematical example of how you generate this. Okay, so first generation is 2 to the 1, which is 8 cells. If you start with 4 cells, start with 4 cells. Second generation, 2 to the 2, which gives you 16. Okay, so you can do these. Usually you can do these by hand. There's a lot of different ways to do it. I like this equation. Okay, are you going to have to, memorize this equation? No, but you should be able to use it. Okay, N stands for the number of cells, so initially, Ni, initial. Okay, so if we start with 5 cells, how many cells will be in generation 4? It's 2 to the N is 4, solve it and you get 80. Okay. This is a fun way to do it if you don't want to do it by hand. Okay? Does that make sense?
Yeah.
Okay. All right. Practice questions. Good. Let's talk about phases of growth. All right. So some of this you should probably already be familiar with because you've been in lab for 8 weeks. Okay? Lag, exponential, stationary, death. Where is metabolism the slowest? Okay, usually lag phase. Why? Those microbes are in shock. Whenever you transfer microbes from one media to another or one living situation to another, just like you, they go into shock. It takes a while for them to get acclimated to their new surrounding. Keep that in mind when you're doing your unknowns. Don't expect them that you've got this beautiful phase, they're in exponential growth phase or stationary phase, and they're doing all this stuff, and suddenly you transfer them and they're going to be just like that. No, they have to go through lag phase. Okay? Now, when they start running out of resources and their environment gets full of toxins, then they go into death phase. Okay? So, when do you want to hit with an antibiotic? Oh yeah, this is where you want to. I mean, this is really when you want to hit with an antibiotic. When do you want to kill with heat? Oh, you guys are too passive. If I want to get them in heat, I've got a bunch of microbes. I'm going to get them right here before they even start. Okay? You can do it in stationary phase, but are you going to have the same results? No. When do you think endospores start to form? Yeah, right at the end of stationary phase. So they're starting, because you don't get endospores all the time, right? When do you get them? When they're stressed. When do they start to be stressed? Right as they're coming into here and they start to decline. By death phase, this is, you've got the same, here it's one in, one out, right? One dies, the other one replaces and stuff like that. But, yeah, so, okay. So good. So you've got all of these things, stationary phase, all right? And watch the video. The video on this is actually really good. It goes through what you expect and what's happening metabolically in all the different phases, okay? So any general questions about this? Okay. All right. So serial dilutions. Okay? I look at a culture like that, it's too crowded in there. You're not going to be able to count colonies. Look at that. There's no way to count colonies and get reliable answers. So you do what is called serial dilution. Okay, so you take 1 milliliter, usually into 9 milliliters of water, so your total volume is 10. Okay, that's key. Students forget that all the time. I did when I was a student. It's not you're dividing by 9, but it's a 10. So it's a 1 to 10 dilution. 1 ml into a total volume of 10. So just remember that that's your total volume that's at the bottom. Okay. And we can use, exponents to do this. But which plate down here would you rather count? Oh yeah, this one is much easier. This one gives you a headache and makes you have Advil. Okay, so this one, you can actually see each colony and you don't have contact inhibition with these, okay? So, yeah. Calculations, okay, all right. This just goes through and it shows you how to do the serial dilutions. And we're looking for the number of CFUs. Have you heard that term before in lab? Okay, colony forming units. So, yeah. And do you just want to do one plate? No. How many plates would be good? 3. Good. Oh, good. Oh, good. So, yeah. So this isn't new. Have you done serial dilutions in lab? We used to do them. You did them? Okay, good. You should do serial dilutions in lab. If not, ask your instructor. So, whose lab are you in? Herrick, yeah, Herrick is on top of, he does that. Yeah, we were students together, so I know we got beat to a pulp routinely. Yeah, so make sure that okay. Ask your instructor, Heidi Ramirez, to do serial dilutions with you if you haven't, because in Herrick's class I know you've been doing it. Okay, so you get some practice. Okay. All right, now what do you do if microbe numbers are too low? This happens. If you've got, clinically this happens. We don't want to see patients that come in and have huge counts of bacteria. We really don't want to see that. Because what does that mean for the patient? Yeah, it means what? It means they're sick, right? So if you're trying to get a culture, oftentimes the numbers are going to be too small, and so there's a couple of ways to get those counts up. Okay, one of them is to filter. So water passes, but the microbes get stuck on the filter, and then you can culture them. When do we do this? This happens every day when they're sampling for bacteria at water treatment facilities. Okay. And so if you're going to go into industry, you decide, hey, nursing isn't for me. I make twice as much money at the poo factory. Okay. You can go into wastewater management. And you'll have to do this because not only do they want to identify, but they want to make sure that they have enough microbes to do metabolic testing on the microbes. Okay. All right. What else can you do? You can put them in the tilt-a-whirl. The test tube tilt-a-whirl. Have you ever used one of these, a centrifuge? Yeah. How many of you used centrifuges before? Okay, yeah, it's a really fun ride. You put the test tubes in, make sure it's balanced, lock it down, and spin it. And what happens is that all of the microbes end up where? At the bottom in some kind of pellet. And you can do this with bacterial cells or human cells or any cells that you're growing in culture. Okay? All right, you can use what's called the most probable number, okay? And this is statistics. So what you try and do is do a dilution series, and you see where you start to get growth and you don't get growth, okay? So I'm not going to give you any MPN calculations because, that's for a more advanced micro class. But as long as you know about most probable number and what you're looking at, okay? And there's some pictures in your book too about what most probable number looks like, okay? And you do do it over a set of 5 tubes, okay? So like up here, you know, we've got 5 positive tubes, we've got 1 positive tube, and then we look at what the series is and we look it up on a chart. I mean, this is really just statistics. And we know about statistics, right? Doesn't always work. Okay, so this is a fun one to use when you're trying to find out and you don't have a lot of cells in your sample. You can do what's called a direct microscopic count. So you have a grid. This is a special microscope slide, and it's elevated up here, and you put your sample here, and there's a grid. And there's a manual that you look at, and you count so many grids randomly throughout here, and then look it up in the manual, and it will tell you how many microbes you have. And so this can work really well if you're talented and you're confident with adding and keeping track of all the grids that are on here. It's hard if you're not very good at microscopy and you get lost in the sauce on the grid and can't add them up correctly. So this really puts in human error. Does that make sense? Yeah, okay. All right, turbidity. What are we doing? We're looking at how turbid or how cloudy a sample is. Out of these 2 tubes, which one is going to have the most microbes? Oh yeah, this one. Yeah, this one definitely. But we can look at that and say that because it's so obvious. This is so, you know, I don't know, overpopulated actually. This won't survive too long. But we have a spectrophotometer that we can use, and we can shine light through it, and so we can tell. And you can do a standard curve with and then compare what you get to the standard curve. Okay, so what other things because this is optics, you can get a lot of interference. What are some of the things that will interfere with this?
Dead cells.
Dead cells, yeah. Can you tell if those are metabolically active? No way, can't really tell, but dead cells will interfere. The other thing that will interfere is if you have a lot of inclusions in your bacteria. So if you've got inclusions that can refract the light or absorb light, you'll get you'll miscalculate very easily. Okay. All right. So if you have a ton of money and a nice lab, like most medical labs do, they have a culture counter, and they use a culture counter, which is a industry standard counter that can count cells. And if it's a fancy culture counter, ours was only like $75,000 or something. It wasn't that expensive. They're at the big hospitals. They have the fancy editions, right? And the culture counter, they can distinguish between all sorts of live, dead, debris, things that electronically, count. Flow cytometry is another way to do it, and this is an even more specific way to get what does your population of your cells look like? How many dead? What percentage? You can tell how many are in mitosis, okay? Or binary if you're dealing with human cells. If you're looking at bacterial cells, you can tell those that are in binary fission. Okay, you can actually measure nuclear material on this. It's pretty cool for a flow cytometer. Okay, all right. Yeah, so this can do living versus dead. What can interfere with the results? If your cells if there's a lot of debris, so anything that is in the media that you're growing your cells with, or the broth that you're growing your cells with, that can interfere. So you have to usually spin down cells and bring them back up and make sure they're nice and clean. Okay? All right. How else can we do this? Okay? This looks like we're looking for aliens, right? I mean, we've got, aluminum and all this stuff. What can we do? Well, we can measure how much metabolic waste the microbes are pushing out. Okay? So we measure gas. Now, this only works if we've got some good gas-producing microbes, right? Okay, because these microbes are going to be doing cellular respiration, so we know we're going to get CO2, but some of them do some other, like for Clostridium, they're going to do other types of metabolism. So they're not only giving off CO2, they're giving off other gases that you can measure, okay? But here's the deal. If we go back to here, let's go back to this. I love this drawing, okay, right here. All right, is every cell going to be in the same metabolic place when you look at a sample? No, no. So that's a problem, because if you're trying to, let's say you catch your culture as they're at the end of the stationary phase and they're about ready to die, you're not going to get accurate results, right? Because their metabolism is slowing down, they're getting stressed. Some of them will be making endospores if that's what they do to survive. And so you won't see this. Yeah, that's one of the things about this kind of measured metabolic activity. Okay, what else can you do? Okay, we can do dry weight estimates. Okay, what we do is we dry the cells on a filter paper usually. Okay, so we know the weight of the filter paper and then we dry it. Is this very accurate? Not really. I mean, this is kind of a funny picture because this balance is meant to measure very heavy things. We can have balances that measure in micrograms, and you might be able to tell some differences there, but it's not the most accurate way to do it because you're picking up debris, you're picking up the mostly dead cells, you're picking up the healthy cells. All of that is mixed in with the dry weight. All you're measuring is biomass, okay? So, okay, yeah, all right. So, have you got the list of all of the different methods we have for measuring? Okay, just make a quick list. I'll turn on your lights so you can go through your notes and make a quick list so you know these are the 5, 6 ways that we talked about measuring. Okay, how many, got your list? Almost. Okay, good. Okay, you got it? All right, so you should have filtration, if they're too low, centrifugation, most probable number, direct microscopic count, turbidity, electronic counting with a Coulter counter or flow cytometer, metabolic activity, and dry weight estimates. Do you have all of those? Good, okay. This will help provide the framework for your nudge sheet, right? To the section on your nudge sheet. Okay, good. All right, any questions about trying to get your microbe numbers up? Okay, all right, let's keep going. All right, so we have all sorts of influences on microbial growth, temperature, osmotic conditions, okay, when you say osmotic conditions, what are you talking about?
Pressure. Not pressure. Water.
Concentration of solutes, exactly, good. Barometric pressure is like how many atmospheres of pressure, okay? Yeah, I was talking to astronauts this week, and actually Sunday night we had our meeting, and we were talking about things that happen in space that do not happen on Earth in terms of cells. And you know, this has to do with barometric pressure. When you're in space, what's your barometric pressure? 0, yeah. And so some of the repair mechanisms that we have in cells stop working at 0G. We have no idea why. Okay? Stem cell differentiation doesn't happen in space. It happens in gravity, but it doesn't happen in space. So if you've got an injury, okay, let's say you got a toothache, and you've got stem cells in your gums and your teeth that are supposed to help heal, or your gut, it's not going to happen. They can't differentiate. Why? That's a golden question. We can't figure out why. So anyway, just things to think about. pH, okay, you know this from lab. Okay, what gases are available, what nutrients, what trace elements and minerals, and what growth factors are in there. And then we have all sorts of biological factors, okay? So we've got, you know, when you look at a microenvironment where there's an infection, it's going to be very different from where it is around in the surrounding tissue. Just like for cancer, you got a tumor, the microenvironment of the tumor is very different than the healthy tissue, okay? So just keep that in mind, especially as we get to ways to control microbial growth, okay? We have 3 cardinal temperatures: minimum, maximum, and optimum. What do you think is optimum temperature for most of the microbes that we're seeing in this class?
37.
37, right, 37, because why? That's where our body temperature is, okay? There are some exceptions. For example, fungus may not grow the best. Some fungi don't grow the best at 37. They like it a little cooler, okay? So each microbe has a minimum, maximum, and an optimum, okay? And if it's optimum, where do they fit down here? What do they belong to? 1, 2, or 3? 2. Mesophiles, right? Those are us. Okay, and that's where most human pathogens are. Okay, thermophiles, they like a maximum temperature. Okay, when you learned about PCR and using Taq polymerase, Taq polymerase comes from a microbe called Thermus aquaticus. It grows in thermal vents, so that would be a good example of a thermophile. And then we have psychrophiles, which grow at very cold temperatures, and they can grow at 0 degrees Celsius. So we look around the planet, you know, just because it's at 0 degrees, there's going to be microbes that are going to take advantage of that and grow, okay? Yeah. All right. So just kind of show you the minimum and maximum, all right? What are your incubators in lab?
37.
Most of them are 37. Some are set a little bit lower so you don't have to come in every day and check on them, okay? All right, halophiles. Halophiles are areas that are, you know, salty, okay? If you've ever flown in to Salt Lake City, you've got the Great Salt Lake there, which has almost, well, it's dwindling down. It used to be, it's almost double what the ocean is. You used to be able to float on it. I don't know, it's really drying out right now. It's a toxic mess. There's a lot of companies that are taking advantage of that, including Morton Salt Company. That's where they get some of their salt. But you fly over and you see all these different colored pools, and it's all the different halophiles that can grow at different salt concentrations, and then they harvest them and use them for, you know, research, sell them for research. So, okay, barophiles. Barophiles are the organisms that can survive under extreme pressure, and if we want to have extreme pressure, we go to the ocean depths to get extreme pressure, okay? So we also have those that really like acidity versus alkalinity versus a neutrophile, which likes around pH 7. Give me an example of a microbe that's an acidophile. a lot of the fungi, what pH do they like? They like around pH 5. That's why they like to grow on your skin, because the oils on your skin make your pH of your skin a little more acidic, and so they like that. Okay. All right, so here's some, minimum, optimum, maximum pHs. Okay, so we've got some that really grow. I wonder where this one got its name. Okay, and then we've got, so anyway, don't memorize this. I'm not going to pull a question off that. Okay, now this you need to know. Okay, you haven't done, have you done glycothiolate tubes yet in lab? Yes, okay, so this is going to be kind of review. All right, when you have aerobic growth, it's going to be near the top of the tube because it likes oxygen. What are most of our pathogens? They're aerobic or they're facultative. Right. Remember, they're going to try and limit anything, okay? Microaerophiles, I would even make this even smaller, okay? And if you do watch the video, the way they show microaerophiles is way too big. It's a very narrow ring in that thioglycolate tube, okay? Aerotolerant and anaerobic, they're going to be all here down in the bottom, okay? So, good. Okay, so you've already done these. How did your tubes turn out? Were they good? Yeah, okay. I hope so. Yeah. What percentage of the atmosphere is oxygen? 21%. Yeah. Mostly nitrogen. We're hearing mostly a nitrogen thing. Okay. I believe you did this last week. Is that right? Or the week before? Oxidase testing? Maybe you do it this week. Can't remember what's your schedule. Okay. All right. What kind of molecule is this? It's an enzyme. Why? ASE, right? Okay. So this oxidase test is great because it shows you if you use oxygen metabolism. Okay, so we look for microbes that have cytochrome oxidase. So they transfer an electron or electron transfer to oxygen. Okay, and if it's positive for oxidase, it turns purple, and if it's negative, then it's doesn't turn purple. Okay, so if we're trying to find the difference between Campylobacter and Neisseria, this is what we do, because Neisseria is positive. Okay, all right, so we've got oxygen. Oxygen is a double-edged sword because as soon as, oxygen is the most electronegative thing that we have commonly in biological systems, and so it's going to want to steal electrons everywhere. So it's not only can do damage to DNA, but it does lipid damage. Okay, so you can get oxidation of lipids, and that's, for a bacteria, they've only got that lipid cell membrane. And once that's damaged, that's kind of done. And so what we do is we have the bacteria have protective enzymes. They have catalase, which you may have seen in Bio 1. Did anybody do this experiment in Bio 1? The catalase and the potatoes and yeah, okay. So catalase is everywhere, right? Okay. So, by the way, if you got a big wound, do you want to put hydrogen peroxide on a big wound? No. How come? Yeah, it can't distinguish between healthy, and what happens is it damages the tissue, so it makes it harder to stitch together. Okay, so yeah, I just listened to an emergency medicine podcast, and they were talking about not using hydrogen peroxide because it actually does a lot of tissue damage. It's better, there's other things. I mean, I don't know. We just pour alcohol and you scream, and that's it. Okay, so catalase, superoxide dismutase, or SOD. Okay, that's another enzyme that what do? We take oxygen, these free radicals, okay, and use superoxide dismutase, and we make hydrogen peroxide and oxygen, and then catalase takes the hydrogen peroxide and converts it to water and oxygen. Okay, these are critical enzymes, and if you get a mutation in one of these and it doesn't work, suddenly your facultative or obligate aerobe is it's gone. Okay. All right, so we've got what are called capnophiles. Capnophiles like high CO2 concentrations. And we actually have incubators and laminar airflow hoods that you can see here, where you work in a sterile environment, but a high CO2 environment, where we grow streptococcus and we grow Neisseria. Okay? And if we don't, if your hospital or facility doesn't have this, they at least have the chambers, okay, that you can put things in and turn on the CO2 and get a high CO2 concentration in there. Okay? All right. Microbial nutrition. What are we doing here? Okay. Cytoplasm, all right, is mostly water. Okay? You've got the macronutrients and the micronutrients. Okay? Micro, needed in tiny bits, manganese, zinc. Yeah, so, okay. I know, I'm going fast, but it's okay. You got all the answers in your book, right? Okay. Oh, by the way, I didn't get any photos from someone. Oh yeah, send me, you've got to do that because I got an email yesterday from the tech people from McGraw-Hill. So send it today, okay? All right, can you help reminder, study buddy? Yeah.
Thank you for bringing that up. A question for you, what edition are you using?
I have two. I have the 9th and the 13th, or the 12th.
Oh,
And I have a preview of the 13th. I don't like it as much as the 12th.
thank you.
Yeah. So, yeah. Okay. So, heterotrophs, autotrophs, photoautotrophs, and chemoautotrophs. We good here? You've had this before. Okay. We're not going to see photoautotrophs and micro. Why? Because they use light. Okay. They're not going to do it. So you should be able to fill this out. Okay. And then chemical factors: hydrogen, nitrogen, phosphorus, and sulfur. Okay. Where do these come from? Rocks on the planet. Okay. Nitrogen. We have a whole nitrogen cycle. Phosphorus come from. We get most of our phosphorus from rock decay. Same with sulfur. Our cells aren't good in converting that mineralized elements into, incorporating them into biological molecules, so we rely on other things like bacteria to do that. Okay, so here's the the micronutrients. Here's the trace elements: potassium, sodium, calcium, magnesium, iron. Okay, you should be able to tell me why each one of these are needed. Why is iron needed?
It's part of hemoglobin.
Yeah, but for a microbe doesn't have hemoglobin.
Probably for structure.
Oh my. Okay. Yeah, you might want to ask yourself, why would a microbe need iron? What does it use magnesium for? Okay, think in terms of enzyme factors, okay? For, you talk about, some of you take a lot of vitamins and supplements and all that stuff. Why are you taking all that stuff? Not really, why? Okay, first of all, they're not regulated, and second of all, we don't know what it is, and number 3, you pee and poop it, most of it out. Okay, so why do you do it? Because what?
Because some work.
Deficiencies. But these are micronutrients. These are micronutrients. Why would you need magnesium? Okay? Think about enzymes. Enzymes sometimes need cofactors in order to work. Okay? This is back to chapter 2 things. Okay? Enzymes aren't just protein. They are mostly proteins, right? But a lot of enzymes need cofactors, and oftentimes these are cations that they need, okay? All right, zinc being one of them, okay? Manganese, magnesium, okay? And they bind to certain places on that enzyme that help it move more efficiently, okay? What happens when you overload your body with a lot of these micronutrients? What happens? Sometimes, but you also want to be careful, okay? Because oftentimes if your cell sees a cation that's 2+, okay, and it can't get calcium for your bones, it might start substituting something else in, okay? Why? They're going to take things that look similar if they don't have it. And we're starting to see this. We see this in populations that are really malnourished, and then they're in an area that their soil has some of these cations in, and their bones aren't as strong because why? They're the enzymes that support the bone growth. Instead of putting in calcium, because that's lacking, they'll find another cation, 2+ cation to put in there. Okay? That's not necessarily a good idea. So for those who like to mega supplement, you might want to rethink that a little bit just from your biochemistry. Okay? Biochemistry point of view. Okay? All right. So growth factors. Okay? These are usually organic acids, some kinds of organic molecules. They're needed in tiny amounts, or they're small amino acids that you can add to cells that will help them grow better. Okay, they're called growth factors. Okay, so sometimes when we do culturing of like human cells, we use tissue extracts. We use actually serum, bovine serum, which came from cows. Okay, why? Because it's got small amounts of this in bioavailable forms. Does that make sense? Just because you throw magnesium in there, magnesium is a hard rock, right? Magnesium is hard. You gotta, if you use a serum, that bovine, that cow has already metabolized it and made it into a more bioavailable form. Okay? So we need everybody. Okay? This is violent. Okay, we need everything, and when you know this from what you've read about the microbiome. I hope you've read a little bit about it, but when you destroy all the members of your community, does anybody do well? No. So yeah. Okay. So the last thing, I think it's the last thing. Yep. The last thing. Wow. We might make it. So you've talked about symbiotic relationships, and under that umbrella of symbiotic relationships, there are mutualisms. What is a mutualism? They both benefit. What is commensalism? Only one benefits and not the other one. And what is a parasitism?
One benefits, the other is harmed.
The other one is harmed. And what is the worst one, which is a parasitoidism? One does well and the other one dies. Okay? That's a parasitoidism. Yeah. Okay. So we've talked about a lot of different kinds of parasites. You've had cards with worms and protozoan on them and things like that. So think about what kind of relationship are most of those. Okay? Okay, we've ecto and endoparasites. We've got intracellular parasites. We've got obligate intracellular parasites. Okay, cool. All right, good. Whew. How'd you do? You okay? That was like a record. Oh my gosh, I hate that. I like to go slow and let's draw and let's talk about it. But I'd rather pack in as much as we can in the time that we have. So you ready for some Kahoot? Okay, let's do some. Because we're going to talk about those questions too, right? So.